Welcome to this video on cells. Cells are the basic units of all living organisms. In A-level biology, we study how cells are structured, how they work, and how they divide. Understanding cells helps us explain processes such as growth, reproduction, and disease. Cell theory and the importance of cells, prokaryotic and eukaryotic cells, key differences. Cell theory states that all living organisms are composed of cells. Cells are the smallest units of life, and new cells arise from pre-existing cells through division. These principles explain how life is organized and how growth, repair, and reproduction occur. For example, when skin heals after a cut, new cells are produced by mitosis to replace damaged ones. Cells also provide a link between structure and function. Specialized cells such as red blood cells are adapted to their role by lacking a nucleus and having a biconcave shape to maximize oxygen transport. Understanding cell theory allows biologists to explain why viruses are not considered living organisms, as they are acellular and cannot reproduce independently. Historically, the development of cell theory was linked to advances in microscopy by scientists such as Robert Hooke and Antony van Leeuwenhoek, whose observations provided evidence for cellular organization. Prokaryotic cells, such as bacteria, are typically much smaller, with diameters around 1 to 5 m and lack membrane-bound organelles. Their genetic material is circular DNA found free in the cytoplasm, often alongside plasmids which carry advantageous genes such as antibiotic resistance. Prokaryotes possess a cell wall made of peptidoglycan and may also have features like flagella or capsules that aid movement and protection. In contrast, eukaryotic cells, found in animals, plants, fungi, and protoctists, are larger and more complex, usually 10 to 100 m in diameter. They contain membrane-bound organelles, including a nucleus with linear DNA associated with histone proteins. This compartmentalization allows greater efficiency and specialization of metabolic processes. For example, mitochondria provide a site for aerobic respiration, while chloroplasts in plant cells enable photosynthesis. Ultrastructure of animal cells, ultrastructure of plant cells. Animal cell ultrastructure refers to the detailed internal structure visible using an electron microscope. Key organelles include the nucleus, which contains chromatin and controls cell activities through gene expression. The nuclear envelope, with nuclear pores, allows the exchange of mRNA and proteins. Mitochondria are double-membraned organelles responsible for aerobic respiration, producing ATP to fuel cellular processes such as active transport. The inner membrane is folded into cristae, increasing surface area for ATP synthesis. The rough endoplasmic reticulum, RER, is studded with ribosomes and is involved in protein synthesis and transport, particularly proteins destined for secretion. In contrast, the smooth endoplasmic reticulum, SER, synthesizes lipids and steroids and plays a role in detoxification. The Golgi apparatus modifies, packages, and sorts proteins into vesicles, often adding carbohydrate groups to form glycoproteins. Lysosomes contain hydrolytic enzymes and are important for intracellular digestion and apoptosis. Plant cells share many features with animal cells but also possess unique structures that reflect their role in photosynthesis and support. One key feature is the cellulose cell wall, which provides structural strength and prevents cell lysis by maintaining turgor pressure. The large permanent vacuole, surrounded by the tonoplast, contains cell sap and plays a role in maintaining osmotic balance and storing substances such as sugars and pigments. Chloroplasts are specialized organelles responsible for photosynthesis and contain chlorophyll within thylakoid membranes. These membranes are stacked into grana, increasing surface area for the light-dependent reactions, while the stroma contains enzymes for the Calvin cycle. Plant cells also contain plasmodesmata, which allow direct cytoplasmic connections between neighboring cells, enabling cell-to-cell communication. Light and electron microscopy, magnification and calibration calculations. Microscopy is central to studying cells. Light microscopes use visible light and glass lenses, with a maximum resolution of around 200 nanometers. They are useful for observing whole cells, tissues, and living specimens, such as onion epidermis or cheek cells. Electron microscopes use beams of electrons and electromagnetic lenses, achieving much higher resolution. Transmission electron microscopes, TEM, allow detailed study of cell ultrastructure, while scanning electron microscopes, SEM, produce three-dimensional images of surfaces. However, electron microscopy requires a vacuum and extensive sample preparation, meaning specimens must be dead. Resolution refers to the ability to distinguish two close points as separate, while magnification is the degree of enlargement. Magnification is calculated using the formula magnification equals image size divided by actual size. Students must be able to rearrange this equation and convert between units such as millimeters, micrometers, and nanometers. Calibration of an eyepiece graticule using a stage micrometer is a required practical skill. This process allows accurate measurement of cell structures under different magnifications. Common errors include failing to convert units correctly or confusing magnification with resolution. Mastery of these calculations supports accurate data analysis and strengthens overall scientific skills. Fluid mosaic model of the cell membrane, diffusion, osmosis, and active transport. The fluid mosaic model describes the structure of cell membranes. Membranes consist of a phospholipid bilayer with hydrophilic heads facing outwards and hydrophobic tails facing inwards. Embedded within this bilayer are proteins, including channel proteins, carrier proteins, and receptors. Cholesterol molecules regulate membrane fluidity, while glycoproteins and glycolipids are involved in cell recognition and adhesion. The term fluid reflects the lateral movement of phospholipids, and mosaic refers to the patchwork arrangement of proteins. Understanding membrane structure allows explanation of selective permeability and transport mechanisms. Transport across cell membranes is vital for maintaining cellular homeostasis. Diffusion is the passive movement of molecules from a region of higher concentration to lower concentration and is influenced by factors such as surface area, concentration gradient, and temperature. Facilitated diffusion involves carrier or channel proteins and is also passive. Osmosis is the diffusion of water across a partially permeable membrane from a higher water potential to a lower water potential. Active transport differs because it requires energy from ATP to move substances against their concentration gradient using carrier proteins. Examples include the uptake of mineral ions by root hair cells and glucose absorption in the ileum. The cell cycle and interphase, mitosis, and its significance. The cell cycle describes the sequence of events leading to cell division and is divided into interphase, mitosis, and cytokinesis. Interphase is the longest stage and includes the G1, S, and G2 phases. During G1, the cell grows and synthesizes proteins. The S phase involves DNA replication, ensuring genetic continuity. In G2, the cell prepares for mitosis by producing spindle proteins and checking for DNA damage. Understanding interphase is important because it explains how cells maintain size and function between divisions. Mitosis is the process of nuclear division resulting in two genetically identical daughter cells, essential for growth, repair, and asexual reproduction. It consists of prophase, metaphase, anaphase, and telophase. During prophase, chromosomes condense and the spindle forms. Metaphase involves alignment of chromosomes at the equator, ensuring equal separation. In anaphase, sister chromatids are pulled apart, and telophase sees the reformation of nuclear envelopes. Cytokinesis then divides the cytoplasm. In this topic, we have explored cell structure, microscopy, transport across membranes, and cell division. Understanding cells provides the foundation for studying all areas of biology. Thank you for watching.